morning. Hello, everyone. Welcome to this Gero Talk. In this occasion, we have the pleasure and the privilege of having the presentation of Lea Grinberg, Associate Professor of the Memory and Aging Center of the University of California, San Francisco. Lea is a medical doctor with a PhD. She has really a huge CV, so I will only mention some aspects, highlighting those most relevant for this Gero Talk. In addition to her academic activity at the university, she also holds positions in other institutions. Just to mention a few: University of Sao Paulo Medical School, Human Proteome Organization, Lawrence Berkeley National Lab, and she's also a member of the Global Brain Health Institute. She also collaborates with professional institutions such as the Alzheimer Association and the Advisory Board of the Latin American Brain Health Institute. Lea has received several honors and awards, the latest being the John Douglas Endowed Chair of the French Alzheimer's Foundation and the honorable mention of the Weill Prize from the American Association of Neuropathologists. The main focus of her research is neurodegenerative diseases. Specifically, the overarching goal of her research program is to map the progression of neurodegenerative diseases and their selective vulnerability from their early pre-symptomatic stages. Today, Lea Grinberg will present us Rewriting the Initial Stages of Alzheimer's Disease, the role of neuromodulatory systems in precognitive symptoms and disease pathogenesis. Okay, Lea, thank you very much. And you can start.
Muchas gracias y buenos días. Un placer estar acá. Yo voy a iniciar la charla ahora. Yo voy a hacer en inglés, que es más fácil para mí. Mismo en portugués, yo creo que no puedo hacer charlas todavía. Pero después vamos a hablar y podemos hablar como quieran. Entonces yo voy a empezar ahora. Déjenme ver si puedo compartir. ¿Pueden ver?
Not yet. Oh, ahora sí.
Bueno, so good morning, and it's really a pleasure to be here. And today I would like to share with you a little bit of my trajectory, rewriting the initial stages of Alzheimer's disease. I think the talk will explain what I mean with it, but more important, and I think more relevant for this audience here, we will be able to talk about what are the clinical manifestations of these initial stages of Alzheimer's disease. So what does it matter in our clinics and what we can do to detect disease in our patients even before the cognitive aspects start to kick in. I will mostly focus on Alzheimer's disease out of all neurodegenerative diseases. And I think for this audience, I don't need to spend a lot of time trying to convince you that Alzheimer's disease is a big problem. At least in the United States, it's estimated that 1 in 8 people age over 55 have Alzheimer's disease, but the costs for treating dementia overall, and Alzheimer's disease is one of the most important, it's even more than if you combine heart disease and cancer together. So there is a big need to find something for this disease. In fact, if we look at the most common causes of death in the United States, Alzheimer's disease is the only one that increased in the last 10 years and increased by a lot. And the reason for this is that despite all the efforts in the field, we don't have a disease-modifying treatment yet. Some may claim that recently we got one approved, but it's to be seen. There is a lot of questions about it. What is important when we try to treat Alzheimer's disease? And some of the critics say that we don't have a drug yet because we did clinical trials in the wrong part of the disease. Neuropathological studies, autopsy studies, teach us that when someone starts showing cognitive decline, the first signs of memory loss, the disease actually is already building up in the brain for many decades, which is this pink square here. And actually, Alzheimer's disease, if we look directly in the brain, it's already building up 10, sometimes 20 years before the first clinical manifestation. So this is what we call prodromal stage, pre-symptomatic stage. There are different ways to call that. And this is the ideal window of treatment, because by the time the symptoms start, the disease is fairly well spread in the brain and many neurons die. We do have improved biomarkers, both in biofluids and in neuroimaging, to detect this pre-symptomatic stage. At single level, they are still not great to distinguish patients from controls because there is some overlapping, but certainly we are getting there, and we give us the opportunity to get to this stage. When we try to develop biomarkers to detect Alzheimer's disease, one of the first questions we need to ask ourselves is, what is the best marker for AD progression? During Alzheimer's disease, different processes happen in the brain, and we would like to be able to measure the one that's more relevant. Every time we think on Alzheimer's disease, I believe that comes to mind two kinds of neuropathological changes. One of them are the amyloid plaques, these deposits of beta amyloid protein outside the cells. They look like plaques, they look like mold in your bread, and they are here represented in gray. The second one is the tau changes, the neurofibrillary changes that we see inside the neurons. This is why they are rounded like this with the nucleus here in the middle. For many years, most of the researchers thinking on Alzheimer's disease, they considered the plaques were the most important change, and everything came after plaques. But actually, when we look at Alzheimer's disease now, we think a little bit different. And why is that? Because in addition to have protein deposits, Alzheimer's disease and other neurodegenerative diseases are characterized by what we call stereotypical spread. So what does it mean? It means that these diseases, they don't show up in any area in the brain in any moment. They tend to always show up in a specific area first, a seeding area, a signature area, ground truth. I mean, there are different names for this. And from there, it will spread in a stereotypical way, in a way that we can predict. So based on this principle, if we understand what is the first areas to show these proteins or neuronal loss, maybe we'll be able to detect where Alzheimer's disease starts. And indeed, neuronal loss and synaptic loss are the best correlate of clinical symptoms. And this is important. We don't need to treat something that doesn't cause any harm, because this is not necessarily a disease. But when we have a process that starts to cause cell death and decline, this is when we want to start treating. So logically, it will make sense to try to detect what is the first area of the brain to show neuronal loss. And this is very difficult. And the reason for this is because each one of us, we have different numbers of neurons in the brain. So even if I get a brain post-mortem, like these two brains I got here, one with Alzheimer's disease with the loss of the superficial layer of the entorhinal cortex, and the other without Alzheimer's disease, and I start to count these neurons, even in the control patient, I don't know if there is no loss there yet, because I have no idea how many neurons were there to start with. We are all different. But I know that protein buildup, protein inclusions, the normal numbers for everybody is 0. So every time I find one, this is already pathology. So back again to Alzheimer's disease neuropathological changes. We have plaques, we have tangles. Which one is more important to detect neuronal death and to detect clinical decline? And the answer to this has been answered by a few papers. I'm highlighting one paper from the Brazilian group of aging studies, or the Brain Bank in Brazil, as many people know it, which is a collection that's population-based. So we had people from all stratus of Alzheimer's disease. We look at almost 1,200 people, and then we created this score based on clinical pathological analysis, giving points to which lesions are more correlated to clinical changes. And the winner here is tangles that we measure by Brack stage. Tangles are not the only neuropathological change that cause problems in the brain. We have hippocampal sclerosis, we have Lewy body, but if you look at this list, amyloid is not here. And indeed, amyloid is not present as a good correlate of clinical decline in any of the series out there. So saying that, I will concentrate most of my talk. We are talking about initial stages of Alzheimer's disease and its clinical correlates in the progression of neurofibrillary tangles or tau changes in the brain. And for those who are very familiar with the field, you may have heard about the Bracken-Brack staging system. So what is this? This is a system we use to stage the tau progression in Alzheimer's disease. Not any tau, not in any disease, tau in Alzheimer's disease. And according to this stage, the first cortical area, and I want to emphasize this cortical area to accumulate tangles, is the entorhinal cortex. Then it spreads to the hippocampus, other limbic and paralimbic areas, association cortex, and finally primary cortex. And this system is very predictable and reproducible. The thing is that when the authors of this system they created, they didn't look at subcortical nuclei. And what I would like to show to you in the next few minutes is that actually subcortical nuclei, especially subcortical nuclei of the neuromodulatory subcortical system, the neurons that produce important neurotransmitters like acetylcholine, serotonin, dopamine, noradrenaline, they are the first to accumulate tau and the first to die. And of course, there are symptoms associated with this. So how did I start these studies? I started studies, as I mentioned before, with the Brazilian Biobank for Aging Studies. So back in São Paulo, Brazil, we have something that's very unique. Not only autopsies are compulsory, so everybody needs an autopsy when they die. Actually, this is the law in whole Brazil, but we have a place to do it. So what happens in Brazil is that despite the fact that autopsy is mandatory, there is no autopsy service everywhere because the government doesn't like to pay for this. But in São Paulo, there is still one. And not only that, it is a very large one. This is an autopsy service that performs about 15,000 autopsies per year. And the reason for this is because all the other autopsy centers in São Paulo closed in the past years. So back in the day when I started my PhD after my residency, I decided to maybe leverage this autopsy service. I got a lot of support, and I wanted to understand what was the threshold between brain normal aging and the early stages of neurodegenerative diseases. So I started to collect brains, and this is something just to show how this autopsy or this collection look like nowadays. As you can see, because it's population-based, most of the donors in our brain bank, now almost 4,000, they are in this category, clinical dementia rating 0. So they had no clinical decline. However, when they come to autopsy, many of them already have sign of neurodegenerative disease because, again, all these diseases had this preclinical stage. And this was the ideal collection to start observing where these pathological changes associated with Alzheimer's disease, and again, here I will emphasize the tau, starts. And here's the tau. This is a histological staining with immunohistochemistry. And when I started to look at these brains, and I was expecting to see tau first in the entorhinal cortex because of the Brack staging, I didn't realize at that time they didn't look at subcortical areas. I started to observe that some cases that didn't have any tau in the entorhinal cortex, they didn't have any amyloid, they didn't have anything in the cortex, they already had tau changes that really look like the ones we see in the cortex. In this area here in the midbrain, this is the substantia nigra, just to orient you, this is the aqueduct where this ball is located. And this area is called dorsal-raphe. So dorsal-raphe is very important because dorsal-raphe produce almost all the serotonin we have in the central nervous system. So I thought, how is it possible? It's really true that the dorsal-raphe is more vulnerable to Alzheimer's disease. And then I started to look at the literature, and I found a lot of papers from the 70s and 80s, and stop in the 90s actually, because 90s when the Brack stage was published, so people felt there was no need to look at this anymore, suggesting that the dorsal-raphe accumulated tau in Alzheimer's disease. And not only this, accumulated early. They didn't know how early, but it seems to be early. So I felt I had the right sample to ask this question. Is it true that the dorsal-raphe accumulates so much more neurons? So I took about 140 cases across all Brack stages, and without knowing the Brack stage, without knowing anything, I look at the dorsal-raphe. And what I found was very interesting. So every case that already had tau in the cortex, starting with Brack stage 1, already had changes in the dorsal-raphe. And then for the almost 40 cases that didn't have any changes in the cortex, about 25% of them, no more, so 75% of them, and they were all older than 50 years of age, they already had changes in the dorsal-raphe. And from this study, we published this paper suggesting that the dorsal-raphe accumulate neurons or neurofibrillary tangles even before the cortex. So as you can imagine, it was very provocative. And they were saying, why a group from Brazil will discover this and no one else discovered this? So we had some problems to publish this paper. However, right after that, a series of papers from other investigators started to come out, inspired by this idea, suggesting that there was enough evidence to suggest that accumulation of tau in the brainstem was very early, and also this new study showing that was even earlier than entorhinal cortex. Even Bracken and his colleagues, they revised their initial cortical stages to include subcortical system. In this case, they look at the locus coeruleus as an earlier stage than Brack1. And actually nowadays, we know that about 15 subcortical structures, including all the cholinergic nuclei, many nuclei in the brainstem and the hypothalamy, they accumulate tau before the cortex and before amyloid is there. And if we think and we examine how we treat symptoms of Alzheimer's disease nowadays, of course, we treat with drugs that will modulate all these neuromodulatory systems. They are not disease modifying, they are symptomatic, but this is what we have nowadays. And it makes a lot of sense. So when we published all these papers that were very provocative, the first question people had, they said, okay, great. So let's suppose that tau really accumulate in the subcortical areas before the entorhinal cortex. So what? If you consider all the neurons that are part of the subcortical system, they comprise less than 1% of all the neurons of our brain. So it doesn't matter at all. We don't have to bother about it. There is no symptoms. There is no harm. So until the neurons get to the cortex, we don't have to bother about it. So we said, okay, this is a very fair question. Let's try to answer that. And to answer that, we need the right tools. Otherwise, we cannot answer these questions. And the right tools in our case was to have this great population from the brain bank in Brazil, because it was population-based and we could look at all stages of Alzheimer's disease. And also employ a way to measure neurons that is unbiased. So in routine neuropathological studies, even in research, we make a cut in a certain structure like here, and then we put in a slide and we count it. But the neurons are not distributed in a homogeneous way in the brain. So in order to understand really what is going on, we have to look at the whole structure to avoid bias. And this is called stereology, and this is what we use there. So we started to get the whole brains. We had the whole structure, so we had to use these very large blocks that I learned how to do during my postdoc in Germany. We had very thick slides, so we could count, and this is exactly what we've done. And these are just some of the 3D reconstructions of the structures we were looking at. So the first questions we wanted to ask is, trying to answer this question, is subcortical tau burden just an epiphenomenon of aging? I mean, it doesn't cause any harm, like you know, some other things that happen during aging with our skin, for instance. And Panos Theophilus, who was my first postdoc in San Francisco and now is an assistant professor, decided to look into this. So we took many cases of people from the age 45 to 80 something. Most of them didn't have Alzheimer's disease. And then we look at the neuronal population in the locus coeruleus, one of these subcortical systems that are first to accumulate tau. So you see, these are 40 cases. They don't really have Alzheimer's disease or any other neurodegenerative disease. And what we realized is that there was no neuronal loss associated to aging, and there was no volume loss. So it told us that if we would detect anything in Alzheimer's disease patients, probably it was not related to aging. It was related to the disease itself. And this is important because, again, with aging, some structures of our body, they change. Maybe it caused no harm, but they change. So one of the biggest problems in this kind of studies is to discriminate between what is aging and what is pathological aging. So we did the same thing with the dorsal raphe, the same one that I detected these first angles years back. And again, we didn't find any change. And Sarah Dunn of this work. And same thing for the substantia nigra. So finally, you know, from these 3 studies, and we have others, we felt, well, it doesn't seem that aging affects this nuclei. So let's see what happened in Alzheimer's disease. And this is exactly what we did. And at that point, when we started doing that, we already had started to work with other groups. And this is work from Udo Rupp that we contributed. And he could show with his group that in BRCA0, so again, no changes in the cortex, different nuclei, subcortical nuclei, I already had evidence of AD tau. And these are showed here in these arrows, and you say, oh, this is just a little bit. Yes, it is. This is the very beginning. By the time the disease is very progressed, then we see all this brown here, we see a lot, but certainly it inspires us. Alex Ehrenberg in my lab, so he started to look at the locus coeruleus again. And then at this point, he detected that when we compare BRCA0 people and BRCA1 people, so kind of the next stage, and we look at the proportion of neurons in the locus coeruleus that had tau, just by going from BRCA0 to BRCA1, we already had the double of the proportion of neurons with tau. So this was a very rapid increase and very early. Eva Larsen, this is a very recent study in my group, is not published yet. She did the same thing with the orexinergic population in the hypothalamus. And the orexinergic population is part of the arousal system, and it's very important to control awakening. And she could show the same thing, that even by comparing BRCA0 to BRCA1, we already have a big change in the number of neurons in this nuclei. Again, suggesting not only that these nuclei accumulate tau very early in the progress of Alzheimer's disease, but this accumulation and the impact that it has is very early too and severe. And these severe changes happen already when pathology in the cortex is just starting. And then we ask this question, okay, we could see this tau accumulating there. So does it cause any harm? Does it kill neurons? Does it cause dysfunctional loss? And we had a series of studies in which we could show that indeed it does. So Panos, again, he looks at the correlation between tau accumulation in this nuclei, locus coeruleus and the dorsal-raphe, and the increase in number of markers that are associated with cell death. In this case here, this first graph, caspase-6, which points to apoptosis, and in this graph here, autophagy with LC3. So you see a line going up here because increased apoptosis, and here a line going down because means autophagy is not working very well, all pointing to this very early effect of this pathology over there. And the same thing if we just simply count cell. In the locus coeruleus, for instance, for every increment of the Brack stage, we already have an 8% loss in volume. All right, so now I want to move to what I think really it's the main message here today and what I would like to discuss with you. I gave this kind of introduction, so I hope it will make easier to understand my point with the next slides, but I would like to talk to you about what is the clinical impact of all these changes. So Lea Grinberg, again, looking at the data from Brazil, he compared what is the odds ratio of having neuropsychiatric symptoms. So again, it has nothing to do with memory changes in people at different Brack stages when we exclude everybody that have any disease that has nothing to do with Alzheimer's disease, confounders. And we got a lot of cases, and we got it because we got this collection from Brazil. So in this particular case, we compare 129 people over 50 with Brack 0 with 200 people at Brack 1 and 2. And just comparing this population that, at very early stages of the disease, we saw that for this increment in disease progression, and again, if we talk about cortical pathology, this is nothing. Most of these people don't even have amyloid yet. We could see a very high odds ratio of some of these symptoms at this point. For agitation, it's about 6 times. And we know that people that will develop dementia, they come to the clinic and they start saying that they got very agitated years before. But also we saw significant change for sleep disturbances, depression, appetite dysfunction, and anxiety. And I'm sure you are very experienced in clinical assessment of people with dementia. You probably will recognize these symptoms in the prodromal stages. People that start developing this and years later, they developed memory. So this is good. This is a good indicator that the subcortical changes, they are already causing clinical problems, but we wanted to look at the biology of this. So not just of this correlation. And this is exactly what we did. And we concentrate a lot in sleep disturbances. And the reason we do it is because there is a high prevalence of sleep disturbances in Alzheimer's disease, but also in other neurodegenerative disease. They often predate the disease-defining symptoms, the cognitive symptoms. And we felt that studying this also, we could learn a little bit about how how sleep function works in humans, because most of the studies come from animals. When we think on the theories explaining sleep disturbances in Alzheimer's disease, the most prevalent is this idea that when we don't sleep well, like if people have sleep apnea, they started to accumulate A-beta in the brain. And the reason is because the A-beta is clean in the lymphatic system during the night when we are sleeping. So if we don't sleep, we accumulate more A beta, and then it becomes a vicious circle because A beta disrupt the sleep-wake circle, especially the delta theta waves, and then we sleep even worse. And this is very well illustrated in this graphic here from Musiak and Holtzman, and they put this idea of the amyloid that's very central, and then tau, for instance, comes later on. However, there are two points that question a little bit this theory. So one of them is what happened with people that have a tauopathy called progressive supranuclear palsy. This is a rare tauopathy that's called Parkinsonism and also other cognitive changes. But what these patients really have early on is what we call hyperinsomnia. They cannot sleep. If we do objective sleep assessment with them, these patients sleep in an average less than 5 hours per night. It's very fragmented, and they cannot compensate in the next day. When we feel tired and we nap during the day, they cannot do it. So despite the fact that people with PSP, they live for years with this hyperinsomnia, they don't accumulate amyloid. So what happens here? So the other thing is that, again, when we look at these nuclei that control sleep and control wake, and they are different nuclei in the brain, and this is from a paper that we published 3 days ago. It's a review that I strongly recommend if you are interested on this. We will see that all the nuclei in the brain that control awake, and they are here in red, they already accumulate tau in Alzheimer's disease very early on. So this line means nuclei that we have evidence that not only tau accumulate early on, but accumulate at track 0. So it tells us, well, maybe what happened is that in normal people, and here just to illustrate, when you have during the day, people stay awake, and during the night, people sleep. And this is because the awake neurons are alert during the day, they are active. And during the night, the sleep neurons start to get active. In Alzheimer's disease, the awake neurons die very much because they are affected early, and we have evidence for this, without affecting that much the sleep-promoting neurons. But in PSP, the disease that they never sleep, probably what's going on is that the awake neurons, they are preserved, they are always on, and the sleep neurons degenerate, so they don't provide this balance. And this is what we decided to test, and Joseph O and Rana Ezer led this study, and we used the same principle that we used before, Stereology. So what did they find? They find that if we look at 3 main nuclei that promote awake, and this is the locus coeruleus, the perifornical nucleus, and we talk about orexin, and the tuberomammillary nucleus that produce histamine. Histamine is important for sleep. When we take Benadryl, we fall asleep as a side effect. So first they look at accumulation of tau. So when you look at controls, and controls are clean because they have no tau, and you compare with Alzheimer's disease, progressive supranuclear palsy, and cortical basal degeneration, which is another tauopathy, you will see that you have significant tau accumulation in all of them. So it almost suggests that for these 3 nuclei, they are equally affected by these 3 diseases. However, as I said, accumulating tau or amyloid or whatever is not important. What is important is if it causes neuronal loss or not. And in this case of these 3 nuclei, we see neuronal loss. And here the controls are darker because it's the number of neurons. If we compare to Alzheimer's disease, we have significant neuronal loss in all these 3 nuclei. But this neuronal loss is less significant in the locus coeruleus in PSP and CBD, and they are not even detected in the orexinergic neurons and in the histaminergic neurons. So in addition to look at this in post-mortem studies, and to make this correlation, we wanted also to try to understand if this tau accumulation in Alzheimer's disease in this nuclei and in other area will have an effect in the alpha wave. And the alpha waves are the ones that are active when we are awake and relaxed. And if they had anything to do with the delta theta synchrony that we know that beta amyloid has. And this was a study published 2 months ago by Kamerini Hanashingi with us. And she could show very clear, a very direct correlation between tau burden and the alpha waves, the one that are there when we are awake, but not with the delta theta waves. So it also suggests to us that indeed in Alzheimer's disease, there is a very strong correlation between this early tau accumulation, loss of neurons in the awake promoting area that will affect the alpha waves and certainly cause sleep problems. So it tells us that this idea that in Alzheimer's disease, sleep problem happens just because there is beta amyloid accumulation. People don't sleep well at night, and then they take naps during the day, or they have excessive sleep during the day happen because they are compensating. But what we propose is not like that. We propose is that there is an effect of the beta amyloid probably, but the effect of tau degenerating the awake nuclei, and then people get tired or sleep during the day, it's even stronger, especially in the early disease stage. Also, this kind of study that seems to be very basic science, it's already affecting the way we deal with sleep in neurodegenerative diseases. And Christine Walsh and Thomas Nyland, who are my partners in crime in these studies, they started this crossover clinical trials with patients with PSP, remember those that don't sleep and it causes a big problem for them. And they are trying to, instead of treating them with zolpidem, which is the most common way to treat these patients and doesn't work, they are trying to give an inhibitor of the orexin, which is suvorexant, that was actually recently approved for insomnia and Alzheimer's disease, trying to shut down the orexinergic system in these patients. And we should have results very soon. So our next steps now, and now almost going to the end, all these patients that we look at, they were follow up in our clinics at UCSF during life. And not only that, they went to polysomnography and other objective sleep studies. So now we are making correlations between polysomnography metrics, clinical metrics, and the neuropathological metrics. And these are preliminary results, but what I can show you so far is that we could show that indeed there is a very strong correlation between changes in staminergic neurons and orexinergic neurons with the sleep. But for locus coeruleus, which is the other awake nuclei, we show a paradoxical pattern. Locus coeruleus is a little bit more complicated. I won't be able to talk much about it now, but it seems to be that we will be learning with this neurodegenerative disease a little bit more, even how sleep is processed in normal humans. And hopefully this will bring us better way to treat the symptoms in Alzheimer's disease. So takeaway of this is that I hope I could show you that the nuclei of the neuromodulatory subcortical system show early vulnerability to AD tau. It does have a clinical impact. I show you a very strong clinical impact with sleep and opportunities to try to treat it. I show you some clinical correlates without too much on the biology of other symptoms that we are exploring right now. But the take-home message is that probably in most of these patients, these first neuropsychiatric manifestations, it's already a manifestation of the disease. This is not a risk factor. This is not a marker. This is already tau, AD tau degenerating this nuclei. And it will bring us a lot of opportunity for treatment, especially when we have disease-modifying treatments for tau to act at the disease at these very early stages. It will not only probably improve the symptoms, but hopefully stop the progress of the disease. If you are interested in reading more about it, I suggest these 3 papers from our group, and I can send them by email later on if someone is interested. And if you are really interested in research in this area, I invite you to join the neuromodulatory subcortical systems with the START, especially for people from Latin America. It's probably free or almost free to join the START with this peer and many others. And I would like to finish thanking all my colleagues and students and my collaborators. This kind of studies take years, they don't happen alone. especially people at UCSF, at the University of São Paulo. And if you are not familiar with GBHI yet, application deadline is September 3rd. Is a wonderful opportunity for training and collaborating and being even more insert with research in Latin America. And thank you very much for the invitation, for your attention, and I will be happy to answer questions.
Okay, Lea, thank you very much. Very interesting conference. I'm a little bit concerned because I haven't been sleeping very well, so I hope I'm not in the prodromal stage. Okay, we have time for some questions. Andrea, then Christian, and then Claudio. I have those hands up. Andrea.
We made the question is in Spanish or in English?
Sí, puede ser como quieras.
Ya, vamos a hacerlo en español para no ser, para ser más fácil. Bueno, primero, felicitaciones con la charla, muy buena, y además creo que para las que es como muy bonita también cómo se construye una historia científica, que creo que de un hallazgo fortuito, después se empieza a construir los puentes para mostrar una teoría. Yo tengo una pregunta en relación a, y que además haya nacido de Sao Paulo, es bien emocionante, tiene una parte de ese proyecto. Bueno, pero la pregunta va a lo siguiente. La teoría de Bracken, basado en la importancia de la hipótesis de la corteza entorrinal, ha llevado todo el desarrollo de pruebas neuropsicológicas para la detección de la enfermedad de Alzheimer y de la importancia de las pruebas de la memoria episódica. Según lo que tú muestras, tendríamos que fijarnos para etapas más prodrómicas en las estructuras subcorticales. Uno de los marcadores sería el sueño, pero existen hipótesis de probables pruebas cognitivas que podrían detectar en etapas tempranas. ¿Qué se sabe? Porque esto nos pasaría de cambiar radicalmente desde la memoria episódica a otras funciones.
Yeah, and I will answer in English because it will be easier for me, but I totally understood. So thank you for your question. And yes, there are. So there is a large group of investigators trying to develop ways to detect early stages of Alzheimer's disease, looking at the locus coeruleus and also at the cholinergic nuclei, so the nucleobasalis of Maynard, in two ways. First, with neuroimaging. So now we have sequences that can measure volume changes in the locus coeruleus. And you may remember that I show you that the locus coeruleus already show about 8% of volume loss only from BRAC 0 to BRAC 1. And this is very similar to the nucleobasalis of Maynard. But also there are lots of evidence that the locus coeruleus is important to consolidation of certain kind of memory, especially the kind of memory in which, after we receive all the information during the day and we have to sort out what will state, what be consolidated, what will throw away. So this, it's very much influenced by the locus coeruleus. So there are many papers in the field already exploring these facts as early biomarkers. And finally, one that's very interesting. So the locus coeruleus affect pupillary response. So there are people trying to develop simple tests that will measure pupillary response, when we throw light in the pupillary, and as a way to look at changes in the locus coeruleus. So locus coeruleus, dorsal cholinergic nuclei are the first. However, I think it's important to mention here, there is something that's very important and cause some issues to early detection at this point. When the locus coeruleus and the orexinergic nuclei start to degenerate, the nuclei that are surviving, they start to overreact, to compensate. And they end up overcompensating because the mechanisms to shut it down are not working very well. So if we measure noradrenaline and orexinergic levels and even metabolites in CSF, we will see an increase and not a decrease in early stage of Alzheimer's disease. And it goes together with increase in RNA expression in the cells that are surviving. So probably biomarkers, they are biofluids, at least for these direct markers, they won't work. Imaging, the pupillary stuff will probably work better. And also clinical things for measuring this kind of memories.
Okay, Lea, thank you very much. Christian?
Gracias por la presentación, súper buena. Tengo dos preguntas. Una tiene que ver con algo más general, y es que un tiempo atrás el consorcio de cohortes de Alzheimer publicó un reporte que dice que al menos en Estados Unidos y en Europa hay una tendencia a la disminución en los casos de Alzheimer, un 13% por década en los últimos 25 años. Y te quería preguntar si tú crees que esto es algo que es una singularidad que está ocurriendo en los países más desarrollados o con mayor prosperidad, o tú crees que es una tendencia natural que deberíamos replicar o deberíamos observar en Latinoamérica, que es un poco la región donde nosotros trabajamos. Y la segunda tiene que ver con esta vulnerabilidad que muestran las regiones subcorticales a la acumulación de tau. ¿Cuál es tu explicación para que estas regiones presenten esta vulnerabilidad a la acumulación de tau?
Okay.
Gracias.
I will start with the second question. There are some theories explaining why these subcortical systems accumulate tau first. So first of all, these are cells that are very large, and they have very long processes. So it means that the metabolic rate of these cells along our life is very high, especially because there are few cells that they have to command the whole brain. So they are more exposed to oxidative stress, for instance, and to have failure in the systems that will get rid of trash, like autophagy and so on. So this disease started to accumulate. The other thing is that many of these nuclei, they are close to the fourth and the third ventricle. And as you remember, we have the brain-blood barrier that will prevent toxins to enter the brain. But these cells are less protected. And the reason for this is because we produce hormones in the brains, and the hormones need to come out. So they come out through vessels in areas of in which the blood-brain barrier is weaker, because the hormones need to get out. And in the same way, they are weaker, so toxins can go in, and they are very close to these areas. So these are the two main explanations. So, but we don't have the answer for sure yet, but good hints. So for your first question, if I understood well, you were mentioning about epidemiological studies showing decline of the prevalence in Alzheimer's disease. Is that correct? Yep. Okay. Yeah. So this is also shown in Scandinavian countries. And I think the best explanations we have for this are two. So first, in clinic, we cannot really detect Alzheimer's disease that even if we can with biomarkers, we cannot exclude other causes of dementia. And dementia, for most part, they have mixed causes. And the vascular component, it's very, very important. Usually, we don't talk a lot about it, but there is a lot of contributions of vascular changes to dementia. So in Nordic countries, in the past decades, there were better public health measures to prevent cardiovascular risk factors like high hypertension and diabetes and obesity. And this influence the brain and influence the vessels in the brain. So I think that has more to do with improvement of this vascular part that also will improve neuroinflammation and maybe even make Alzheimer's disease progress in a lower pace than the Alzheimer's disease itself. So what I mean is that I think it's important to make this distinction between dementia, even if we call dementia because of Alzheimer's disease, it's never pure, and Alzheimer's disease biological processes itself. Does it make sense?
Yeah, absolutely.
So in Latin America, we have a lot to do because, you know, cardiovascular risk factors are not well controlled. Yeah.
Thanks so much.
Claudio?
Thank you, Lea, for this beautiful and inspiring presentation. So thinking on the translational potential, so there are many models to study tau pathology in mice. Also now we have the spreading models that resemble more the sporadic cases. So is this early accumulation of tau, observe in any of these models to at least try to study why this is happening? It's really interesting what you said, that these neurons are long, because in ALS and Parkinson, at the end, it's the same problem. These cells have a particular metabolism. So it's quite interesting what you said. Thinking on stopping the spreading, stopping the spreading of the pathology.
Right. So yeah, thank you for this question. That's very important also. So you mentioned that there are many animal models for tau spread, and indeed they are, but I think I have to disagree with you in the sense that this tau spread is Alzheimer's disease spread. Most of these experimental model, at least in my knowledge, they mimic better tauopathies and primary tauopathies. I'm not really familiar with a case that animal models that really have AD tau. However, there is a colleague called David Weinschenker, and he published a paper a few years ago in Brain that's very interesting. They use rats that have the APPPS1 mutation. So this is a mutation for amyloid, right? Not for tau. So usually these experimental models that have amyloid changes, they don't show tau changes, right? Which is a problem in Alzheimer's disease. But the rats, they can have tau changes as opposed to mice. So in this model that he studied, the changes of tau in the locus coeruleus is the first change of tau in these rats. And it happened at the same time that amyloid start to accumulate. So, which is around 3 months of age. And then from there, this tau in these rats, they move to entorhinal cortex and hippocampus. So this is one of the only experimental models I know about tau. But there is something that I think is very interesting, and we could never show in humans because there is no good method for this, but also have to pay attention. So in all these mice model, and now I'm talking about mice, not rats, that have amyloid mutations, any one of them, I mean, they were tested more than 10. If you ablate the locus coeruleus, you can use optogenetics or virus, whatever you want. What happened is that you have an explosion in the number of plaques in the cortex. And then when you rescue the locus coeruleus, and again, you can do it because you use these reversible, schemes, the number of plaques go down. And the reason for this is because the locus coeruleus also control microglia function via a gene called ADAM10, which shows up in GWAS studies in Alzheimer's disease. So even if it's not the tau itself, just is spreading, right, and causing the disease, at least in animal models, even by damaging the locus coeruleus itself, it will already have an effect in the amyloid accumulation in the cortex. There is a more recent study from a group showing how these changes in locus coeruleus also will affect receptors, noradrenaline, especially I think it's the alpha-2 receptors in the brain. And these receptors, when they encounter beta amyloid oligomers, they started to malfunction. And what we will have is an increase of tau because of changes in GSK3. So, we are learning more and more that it's not only the spreading or how the disease gets to the cortex, but even by ablating these nuclei, you start having problems in cortical areas by proxy, so to say. Still a lot to study.
Yes, thank you.
Gracias. Felipe.
Yes, thanks, Lea, for that wonderful talk. Yeah, it's quite interesting what you said about this characteristic of neurons that have like long extensions, and also that they pass close to these fenestrated capillaries, right? That they are more exposed to the periphery. There's any evidence that axons or these, start to degenerate early? That's one question, because, or if these kind of these axons passing close to the fenestrated capillaries are usually more kind of negative for other kind of neurons. It's kind of a general thing that's happened, and those are my two questions, basically.
Yeah, thank you for your question. This is something we are not very good in looking neuropathological studies because we are looking at these sections. But yes, there is evidence both in Lewy body disease and in Alzheimer's disease that the generation of the neurons, they start earlier than the soma, the body of the cell. And then we said these threads or Lewy neurites in Lewy body. Ideally, we would like to trace them. So there are some groups, it is very difficult to do and it's time consuming, that they use light sheet microscopy. So it's this technique that you can clear the tissue and instead of section, you can stain and then do 3D reconstructions in subcortical nuclei in Alzheimer's disease. There is a investigator called Saba Dori, and he's at Stockholm, and he has very interesting results for locus ceruleus and orexinergic neurons. So the orexinergic neurons, they innervate the locus ceruleus, and he can show in early disease stages that these processes from the orexinergic neurons getting to the locus ceruleus, they degenerate very, very early. And this is just one of the evidence that probably there are many others if we look closely.
Thank you very much.
Sure.
Ok, estamos prácticamente en la hora, a lo mejor hay tiempo para una última pregunta. Si nadie tiene... bueno, ya, te la concedo, Claudio.
Cortita, tú mencionaste esta respuesta en lo anterior, pero sería súper interesante, probablemente son muy pocas neuronas para hacer algún método de imaging y diagnóstico, porque dados los porcentajes sería, no sé, interesante pensar en salud pública, hacer screening en la población temprano.
Yeah.
¿Hay alguien trabajando en esto?
Yeah, there are many people working on this, and I have 2 R01s on this actually. So the idea is to improve the methods we have to detect volumetric changes, not the neuronal loss, because this we don't have resolution with imaging, but for volume we might have. And there are many people working on this. If you write to me, I can send you some papers. And this is the idea, to create a clinical sequence for MRI that will be very cheap to screen the population, because we all have different volumes. The idea will be to do like colonoscopy, you get, your sequence at the age of 40, 50, whatever, depending on the risk score. And then you come back in 5 years and see what is going on. So this is the idea. I don't know if it will work, but many people working on this and seem to be going in the right direction.
Lea, solo lo último, que a 1 minuto, así que no puedo dejar de preguntarte. Tú mencionaste que la frase prodrómica sería como el ideal para poder implementar algunos tratamientos, ¿no es cierto? Mencionaste, me da la impresión, algo durante tu conferencia, pero si pudieras hablar brevemente de qué tipo de tratamientos contamos con actualmente, si pudiéramos intervenir en esa fase.
Okay, so actually we don't have anything that has proved to be disease modifying, but I would say that even treatments, there are now many tau treatments in clinical trials. Some didn't work already, but maybe some will work. So I would say that this will be a good way because we know that the change is caused by tau. But I will say, because of this experimental study showing that the degeneration of these nuclei affect the neuroinflammation and what happens in the cortex, I would say that really more tailored symptomatic treatment, trying to compensate these neurotransmitters, considering that they overreact and so on, might be, a way to at least delay disease progression. But I mean, this is theory, it has not been tested.
Ok, muchas gracias, Lea. Bueno, estamos en la hora. Por supuesto, podríamos seguir haciendo muchas preguntas porque fue una presentación muy interesante, pero en aras del tiempo solo cabe agradecerle nuevamente que hayas estado con nosotros hoy, que hayas madrugado, tienes muchas horas menos allá, así que muchas gracias, muy interesante. Aprovecho de despedirme de todos y todas y nos vemos en la próxima Gero Talk.